We just finished up with the activation of CD4 plus T helper cells, and definitely the best way to activate these cells would be the interaction of the helper T cell with the T cell receptor to the antigen presenting cell. And so here are the different types of T helper cells that are differentiating out of that activation. So be Th17 cells, they produce interleukin 17 and contribute to inflammation. Th1 cells, they contribute to IF and gamma, which activates macrophages. Th2, they activate B cells, and that's actually important to activate antibodies. They also produce IgEs to activate eosinophils. So here would be the different types of activations of other parts of the immune system as triggered by helper T cells. And you can definitely see that you are activating more neutrophils that are participating in an inflammatory response. You are activating mast cells and basophils and eosinophils that will help with eukaryotic parasitic infections such as worm infections. Then you are enhancing the activity of macrophages that become antigen presenting cells, and we'll start all over here, these antigen presenting cells. And then you have intracellular bacteria and protozoa that will be attacked by these T cells. We also have regulatory T cells, and the abbreviated Treg. That's a subset of the CD4 plus cells. They carry an additional CD25 molecule on their surface as a recognition receptor. They suppress T cells against self, and that means they protect intestinal bacteria required for digestion or protect the fetus. So that means that's your protective measure, that you don't attack something that's actually good for you, like your microbiota. Let's move on to CD8 plus T cells. Those are cytotoxic T cells. They're activated into cytotoxic T lymphocytes with the help of helper T cells and other co-stimulatory signals. Cytotoxic lymphocytes, they recognize and kill self cells altered by infection. Self cells carry endogenous antigens on a surface and presented with MHC class 1 molecules. And here's important: see, cytotoxic T lymphocytes, they release perforin and granzymes that induce apoptosis. That's a fancy term for cell suicide or induced cell death, and typically a virus-infected cell. Here's a diagram that shows you how cytotoxic T cells can kill virus-infected target cells. So here would be your virus-infected target cell, and now we have the interaction of a helper T cell with this virus-infected cell. So this interaction is really important because now this T cell is figuring out a strategy how to get rid of this antigen that is displayed here in this MHC class 1 molecule. Once the T cell has figured out what to do, it will induce whatever it takes to get rid of the cell. And so in this case, we are targeting an infected target cell, virus-infected cell, and kill it before it can spread the virus. Cytotoxic T cells, they can induce something called apoptosis. That's definitely a term that you should know. Apoptosis is programmed cell death, and that means basically you're inducing the cell to commit suicide. All of the cellular components will be basically shredded, and then the cell implodes and disappears. So apoptosis is a mechanism of getting rid of dangerous cells, like virus-infected cells, cancer cells, and some other cells. It's referred to as programmed cell death. It prevents the spread of viruses into other cells, and also if a cell is induced, the apoptosis induced, it will basically shred the genome into fragments, causing the membranes to bulge out and pebble, basically. So here is an image showing apoptosis. This cell here is going into apoptosis. This is a normal cell. Now let's move on to extracellular killing by the immune system. And so that will be the job of the natural killer cells. Think of them as snipers of the immune system. They will go after granular leukocytes that destroy cells that don't express the MHC class 1 self-antigens. They will kill virus-infected and tumor cells and attack parasites. They're not always stimulated by an antigen, but they form pores in the target cell, leading to lysis and apoptosis, just like we had with the complement system and the membrane attack complex. So here is a summary of cells that function in cell-mediated immunity. You have all of these T cells, helper T cells in various forms right here. Cytotoxic T cells, regulatory T cells, and you've activated macrophages and natural killer cells. And so you can use this chart right here, this table, to study. Now let's take a look at antibody-dependent cell-mediated cytotoxicity. For example, worm infections, protozoas or helminths that are too large to be phagocytized by a single cell. So what are you going to do to get rid of a pinworm infection? So basically the strategy is to coat the protozoan or the helminth with antibodies. Then the immune system cells attach to the variable regions of these antibodies, and then the target cell is lysed by chemicals secreted by the immune system cells. So basically you're trying to drill a hole into the surface of the pathogen. In this case it's a whole animal like a worm, and then try to leak out the contents. So here's an example of this antibody-dependent cell-mediated cytotoxicity. So here we have a macrophage, and we have a brave eosinophil that's trying to get, fight with this worm infection right here. And here would be a more realistic view down here of this pathogen that's, as it is, engulfed with eosinophils that are dropping enzymes and whatever stuff that is in their granules. They will drop it on top of the pathogen until the pathogen is dead. Now let's take a look at immunological memory. The first response is always more difficult because you're trying to figure out how to make an antibody, for example, against a specific enemy. Once you have that antibody, you don't have to worry about it that much anymore because you're keeping memory cells around. And once you see the same pathogen again, the memory cells will be very quickly reactivated. They turn into plasma cells and they churn out antibodies. So the secondary immune response is stage after second exposure to the antigen. It's much faster. It lasts many days and has a much greater magnitude. And it's because memory cells are being converted into plasma cells and churning out antibodies. The antibody titer is the relative amount of antibody in serum. It reflects the intensity of the humoral B cell mediated response. And then here you have the IgM is produced first, followed by IgGs. That's fine. So here would be your primary and secondary immune response to an antigen. I would like you to follow the blue line. So first response is production of an IgG. So it takes 2 weeks here. That's why they tell you it will take 2 weeks after you get your flu shot before you have the immunity that they want you to have. Now, a secondary response right here. The pathogen hits, and then right away the memory cells are converted into plasma cells, and now you have a much better, much faster response. Now let's review the types of adaptive immunity. So you have 4 types here. Naturally acquired active immunity, as is in resulting from an infection. So you got sick and then you eventually learned how to make the right antibody. Naturally acquired passive immunity, that will be across the placenta or via colostrum for newborns as they are drinking mother's milk, or even before they were born, transplacental. Artificial acquired active immunity, that will be artificial, refers to the vaccination, and it's active immunity because you're learning to make the right antibodies. So vaccination, and again, that is really the way to go. That is the best way to go, that you're showing your immune system what's dangerous, and then you are teaching it to how to make an antibody. And then artificially acquired passive immunity, that means you get an injection of IgGs or antibodies. Now let's take a look at adaptive immunity. It could be either naturally acquired or artificially acquired. Naturally acquired adaptive immunity could be active or passive. In the active case, the antigen enters the body naturally, and the body produces antibodies and then specialized lymphocytes. Passive means that the antibodies pass from the mother to the fetus by placenta or infant via mother's milk. So it means the infant never learned how to make the antibody, just got it externally. So passive means the body never learned how to make the antibody, it just got it from some external source. In this case, it will be natural. That means through mother's milk, for example. And on the artificially acquired, we also have active and passive types of immunity. Active means that you got vaccinated, basically. So artificially acquired means you got vaccinated. You're introducing a weakened or dead pathogen into the body, and then the body produces antibodies, and now you're going to have those memory cells around. Passive would be you got preformed antibodies from some external source, some anti-venom or antitoxin maybe, or some other injection that gives you the antibodies, but that's temporary. For example, they are talking about it in the news that they are using the serum from people that recovered from COVID-19 and using the IgGs that they have produced, these antibodies. This would be passive immunity. If you're injecting another patient that's going through the disease right now, and you're injecting them with plasma that contains the antibodies, that would be a typical example of artificially acquired passive immunity. Here are the types of adaptive immunity. So in interserum, those would be blood-derived fluids containing antibodies, and serology is the study of reactions between antibodies and antigens, and globulins, those are serum proteins. The most important fraction of these globulins will be the gamma globulins. That would be the serum fraction containing antibodies. And if you were to run these different antibodies on a gel, you'll be able to separate out the different globular fractions. And here are the globulins. Here's where you loaded them, and then they separated out as they migrated toward the positive pole, and you have albumin, relatively small, and there are different types of globulins. And here's the summary slide of humoral versus cellular immune functions. Humoral entirely hinges on B cells, and B cells make antibodies. They have to go through a clonal selection procedure, which takes about a week to make the right antibody, but in the end, they are making antibody. If they're secreting antibodies actively, then that will be plasma cells. If they are inactive and just keeping a sample of their clone of B cells around, that will be memory cells. So humoral immunity hinges entirely on B cells and the antibodies that they produce. Cellular immune functions, that is a variety of different cells. I call it the man-to-man or cell-to-cell combat, and so they will engage in cell-to-cell combat. And here is the interaction, is critical interaction of cytotoxic or T lymphocytes interacting with an antigen presenting cell right here, and then making a strategy of how to combat the infection or the disease that's afflicting you, and they can activate other branches of the immune system.